Hello everyone, welcome to my presentation. This is Xiaofeng, and I'm gonna talk to you about neurotransmission in this video. Neurons, also called nerve cells, are the fundamental units of the brain and nervous system. The cells responsible for receiving sensory input from the external world, for sending motor commands to our muscles, and for transforming and relaying the electrical signals at every step in between. There are actually three kinds of neurons. They are each as sensory, motor, and interneurons. Just as the picture shows, a neuron is a tree. A neuron has three main parts: dendrins, an axon, and the cell body, so-called soma, which can be represented at the branches, roots, and the trunk of the tree. A dendrin is where a neuron receives input from other cells. Dendrins branch as they move towards their tips, just like tree branches do, and they even have leaf-like structures on them called spines. The axon, which you can imagine as a tree root, is the output structure of the neuron. When a neuron wants to talk to another neuron, it sends an electrical message called an action potential throughout the entire axon. The soma, which is a cell body, and you can think it like a tree trunk, is where the nucleus lies, where the neuron's DNA is housed, and where proteins are made to be transported throughout the axon and dendrins. And synapse is the chemical junction between the axon terminals of one neuron and the dendrins of the next. It is a gap where specialized chemical interactions can occur, rather than the actual structure. Let's look at the action potential. A large enough depolarization event, perhaps resulting from multiple depolarizing input that happen at the same time, can lead to a production of an actual potential. An action potential, unlike a graded potential, is an all or none event. It may or may not occur, but when it does occur, it will always be the same size. An action potential begins with the when a depolarization increases the membrane voltage, so that is the crosses the threshold value around like 55 minus. After a short time, sodium channels self-inactivated, close and become unresponsive to voltage, stopping the influx of the sodium. A set of voltage gated like potassium channels open, allowing potassium to rush out of the cell down its electrochemical gradient. These events rapidly decrease the membrane potential, bringing it back towards its normal resting state. The voltage-gated potassium channel stays open a little longer than needed to bring the membrane back to its resting potential. The results in the phenomenon called on the shot, in which the membrane potential briefly dips lower than its resting potential. Eventually, in the end, the voltage-gated potassium channels close and the membrane potential stabilized at the resting potential. The sodium channel returns to their normal state, remain closed, but once more becoming responsive to voltage. The action potential cycle may then begin again. Here are some terms about action potential. Action potential is a short-term change in the electrical potential that travels along a cell. Depolarization, a decrease in the difference in voltage between the inside and outside of the neuron. Hypolarize is to increase the polarity of something, especially the polarity across a biological membrane. Node of Ranvier, a small constriction in the myelin threads, of axons. And saltatory conduction, the process of regenerating the action potential at each node of review. The process of a neuron that it's communicated with each other is actually complex. There's a picture on the left showing approximately how it's done. The synapse, like neuron communicate with one another at junctions called synapse. At a synapse, one neuron sends a message to the target neuron, which is another cell. A single axon can have multiple branches, allowing it to make synapses on various postsynaptic cells. Similarly, a single neuron can receive thousands of synaptic inputs from many different presynaptic sending neurons. Inside the axon terminal of the sending cell are many synaptic vesicles. These are membrane-bound spheres filled with neurotransmitter molecules. There is a small gap between the axon terminals of the presynaptic neuron and the membrane of the postsynaptic cell. And this gap is called the synaptic cleft. When the action potential or a nerve impulse arrives in the axon terminal, it activates voltage gate calcium channels in the cell membrane. The molecules of neurotransmitter diffuse across the synaptic cleft and bind to the receptor proteins on the postsynaptic cell. Activation of postsynaptic receptors leads to the opening of or closing of ion channels in the cell membrane. This may be depolarization, make the inside of the cell more positive, or hypolarization, make the inside of the cell more negative, depending on the ions involved. Thank you for watching this video.